Welcome to the deep dive. I'm genuinely excited today. We're going to jump into something truly foundational. Our mission, really your mission if you choose to accept it, is to go on a comprehensive deep dive into the fundamental principles of cell and membrane function. We're not just aiming to skim the surface here. We're going to pull apart the layers, connect all the dots, and really help you understand how our billions of cells actually work. You know, from their internal machinery to how they constantly interact with their environment. The goal: by the end of this, you'll have a grasp of this information like the elite professionals in this field.
And that's exactly it, isn't it? What's just completely fascinating is understanding how these seemingly tiny microscopic details dictate the grand functions of life itself. I mean, every single process in the human body, think about it, from a simple thought to complex movement, it ultimately boils down to this intricate dance happening right at the cellular level. So getting a real mastery over these core principles, that's the foundation for comprehending, well, all of physiology.
Absolutely. And get ready for some serious aha moments, I think. We'll be breaking down the eukaryotic cell's components, really dissecting the plasma membrane structure and its crucial gatekeeping role. We'll explore the incredible variety of transport proteins, uncover the fundamental forces driving molecular movement like those really powerful electrochemical gradients, and finally we'll get into the essential properties of the solutions that constantly bathe our cells. So let's start our journey right inside the cell itself. Okay, so we're starting with the basic definition. A eukaryotic cell, its defining feature, what really sets it apart, is that membrane-delimited nucleus. Now, just a quick note, there are a few exceptions in the human body, like mature red blood cells and also cells in the lens of the eye. They actually don't have a nucleus, which is interesting.
Right. So conceptually, you can think of the eukaryotic cell as being divided into, well, two main compartments. That nucleus we just mentioned, and then everything else, the cytoplasm. And the cytoplasm itself is basically an aqueous solution, but it's just incredibly packed with numerous organelles, and each one has a highly specialized role.
It really is like a miniature, super efficient factory, isn't it? Each department contributing. So let's take a quick tour through these key cellular components and what they do. First up, the cytosol. Think of this as the main workshop floor. It's where a lot of the cell's metabolism, its chemical reactions happen, and it's also where protein synthesis occurs, thanks to those free ribosomes floating around.
And giving that workshop its structure, its shape, is the cytoskeleton. It's the cell's internal scaffolding, providing shape, sure, but it's also dynamic. It's a transport network enabling cell movement and facilitating all that crucial intracellular transport, moving things around inside.
Then we have the nucleus. This is the control center, really. It houses the human genome, that's the 22 autosomes and the 2 sex chromosomes, and it's the site where all the DNA and RNA synthesis takes place, all the blueprints.
And right from the nucleus, we move to the energy producers, the mitochondria, the famous powerhouses, right? They synthesize ATP through oxidative phosphorylation. But they're not just about energy, they're also critically involved in storing calcium ions, K2+ wire.
Right. And speaking of storage and synthesis, the smooth endoplasmic reticulum, or SER, is involved in making lipids and, yes, also storing more K2+Y. Now, remember those free ribosomes in the cytosol we mentioned? They're specifically translating mRNA into proteins that are going to stay right there in the cytosol. But if a protein needs to be embedded in a membrane or maybe secreted out of the cell, its mRNA is handled by the rough endoplasmic reticulum, the RER.
Exactly. It's a division of labor, even within protein production. Then we've got the cell's recycling and waste management system, the lysosome. It's basically the degradation center for intracellular waste. The endosome, on the other hand, acts as a critical hub for cellular uptake. It's involved in bringing things in, like cholesterol, removing receptors from the plasma membrane, taking up small molecules and water, and even internalizing large stuff like bacteria or cell debris.
And once proteins and lipids are made, they often head to the Golgi apparatus. This is really the cell's packaging and shipping department. It modifies, sorts, and packages proteins and lipids for delivery elsewhere, either to other organelles or out of the cell entirely. We also have other specialized degradation systems, the proteasome, which specifically breaks down intracellular proteins, and the peroxisome, which focuses on detoxification.
So when you look at all these parts together, the really profound thing is how this organized division of labor allows cells to perform incredibly complex functions so efficiently. But it does raise a really important question. How do these different compartments, and the cell as a whole, actually communicate and move things around, especially across that critical outer boundary?
That's a perfect transition. Let's talk about that crucial outer boundary, the plasma membrane. It's this incredibly thin, dynamic layer, only about 5 nanometers thick, separating the cell's inside world from the outside environment.
And it is so much more than just a wall. The membrane has several critical functions. First, yes, selective transport, precisely controlling what gets in and out, mainly via those membrane transport proteins. Then, it's vital for cell recognition, using surface antigens to identify other cells. It's also key for cell communication, hosting receptors for neurotransmitters, hormones, triggering signals inside the cell. And finally, the plasma membrane is absolutely crucial for tissue organization. It forms cell junctions, both temporary and permanent, and interacts with the extracellular matrix through cell adhesion molecules. It holds tissues together.
Wow, so it's a gatekeeper, a communicator, and an organizer. Let's really dive into its structure and composition because, like you said, it's not just a simple barrier, it's sophisticated. At its core is the lipid bilayer, mostly phospholipids and phosphoglycerides. And the key to its structure is their amphipathic nature. They have a charged or polar head, hydrophilic, loves water, and two nonpolar tails, hydrophobic fatty acyl chains that, fear water. This dual nature is absolutely vital. The hydrophobic tails spontaneously hide together on the inside, away from water, while the hydrophilic heads face the watery environments both inside and outside the cell. That's how the bilayer forms.
And within these lipids, you find different types of phospholipids. Common ones have a glycerol backbone. Think choline, ethanolamine, serine, and acetylglycerol. Then there's sphingomyelin, which uses a sphingosine backbone instead. But what's really fascinating is their asymmetrical distribution. It's not random. Phosphatidylcholine and sphingomyelin, they tend to hang out on the outer leaflet facing outside, whereas phosphatidylethanolamine, phosphatidylserine, and especially phosphatidylinositol are mostly on the inner leaflet facing the cytoplasm. And that phosphatidylinositol plays a really key signaling role right there from the inside.
And we can't forget cholesterol. It's a huge player, making up maybe 50% of the lipids found in both leaflets. It's absolutely essential for stabilizing the membrane, especially at our body temp, 37 degrees C. Keeps it from being too fluid or too rigid, just right. We also find minor things like glycolipids with carbohydrate heads, including GPI, which is used to anchor certain proteins to the membrane. Now, despite all the structure, the membrane is incredibly dynamic. It has membrane fluidity. Lipids and proteins are constantly moving laterally, diffusing within the plane of the membrane. This fluidity increases with temperature, and also if there are more unsaturated fatty acyl chains, those introduce kinks in the tails that prevent tight packing, allowing more movement.
But even with that fluidity, movement isn't totally free. It can be constrained, and the membrane can form specific domains. For instance, some proteins get anchored to the internal cytoskeleton, which limits how far they can wander. And then you have highly organized structures like junctional complexes, think tight junctions in epithelial tissues. These literally seal off parts of the membrane, creating distinct apical and basolateral domains. This separation is absolutely critical for something called vectorial transport, moving substances in a specific direction across the cell layer. Super important for organs like your gut and kidneys. And one more thing, lipid rafts. These are like little dynamic platforms, aggregations of sphingomyelin and cholesterol that float around. They associate with specific proteins, often grouping signaling molecules together in functional microdomains.
Right. So connecting this back, the plasma membrane isn't just a passive barrier at all. It's an active, dynamic participant in everything the cell does, constantly adapting, organizing itself, a truly intelligent border. Okay, now this is where it gets really interesting. How does the cell actually move things across that intelligent border? Our sources point out something amazing. About 10% of all human genes, maybe 2,000 genes, code for these crucial membrane transport proteins. And they're also targets for a huge number of drugs. So let's explore the 4 major classes of these specialized gatekeepers.
Yeah, and just to give you a sense of their speed, their efficiency, pores, which are basically always open, can move something like 109 molecules per second. Incredible. Channels, which are gated, open and close, they operate around 106 to 108 molecules per second. And then the sloop carriers and ATP-dependent transporters, they work through binding and shape changes, more like a revolving door. Their rates are slower, maybe 102 to 104 per second, but even that slow rate is incredibly fast when you think about the scale. These things are workhorses.
Truly impressive numbers. Okay, let's dive into each type, starting with water channels, also known as aquaporins or AQPs. These are the main routes for passive water movement. Yes, water can sneak through other ways, but AQPs are the highways, allowing rapid water flow. They're found all over brain, lungs, kidneys, GI tract, liver. And cells can have different types, different isoforms. Some, like kidney collecting ducts, even have multiple types together, AQP2, AQP3, AQP4.
And what's really vital there in the kidney is the amount of AQP2, specifically in the apical membrane, the side facing the urine, is tightly regulated by antidiuretic hormone, ADH, also called arginine vasopressin. This hormonal control is absolutely critical for your kidney's ability to concentrate urine and maintain your body's overall fluid balance. It's also worth noting some aquaporins, called aquaglyceroporins, are a bit less specific. They let water through, sure, but also small uncharged molecules like glycerol and urea. And mostly, how they're regulated is by changing the number of them in the membrane, adding more when needed, taking them away when not.
And just a quick look at their design. Each AQP molecule has 6 domains that span the membrane, forming a central pore for water. But then 4 of these individual AQP monomers actually cluster together to form a tetramer in the membrane, and each monomer within that cluster functions as its own independent water channel. Very efficient design.
Okay, next up, ion channels. These are in all cells, but they're especially critical for excitable cells, neurons, muscle cells where you need rapid electrical signals. We classify them in a few ways. First, by selectivity. Some are highly specific, maybe only letting potassium through, while others are less picky. Second, by conductance. How fast ions flow when the channel is open. This can vary too. For instance, an inward rectifier channel might let ions flow in more easily than out.
And crucially, they're classified by their gating mechanism, how they open and close. Because they don't just stay open, they flicker, spontaneously fluctuating between open and closed states. And their gating can be controlled very precisely by different things. Changes in the membrane voltage, binding of specific molecules outside the cell, like acetylcholine at the neuromuscular junction, messengers inside the cell, like K++ ATP, cyclic GMP, or even physical stretching of the membrane. Like aquaporins, you can regulate them by changing the number of channels or by controlling their gating, how often or how long they stay open. And the sheer speed is mind-boggling. A single open channel can let through something like 12.5 million ions every second.
Moving on, we have the solute carriers, or SLCs. This is a huge, huge family, over 50 groups, nearly 400 identified transporters. We group them into 3 main modes. First, uniporters. Simple. They transport just one type of molecule across. The classic example is GLUT1 for glucose transport.
Then you have symporters, also called cotransporters. These couple the movement of 2 or more different molecules, all moving in the same direction. A really key example is the Na plus K plus 50Cl symporter, often called NKCC2. It's absolutely vital for the kidney's ability to dilute and concentrate urine. Shows how these transporters fit into bigger physiological jobs. And finally, antiporters or exchange transporters. These also couple movement but in opposite directions. One molecule goes in, another goes out. The Na plus H plus antiporter, specifically NHE1, is found in pretty much all cells and plays a huge role in keeping intracellular pH stable.
Okay, and our 4th major class, the ATP-dependent transporters. As the name says, these guys directly use energy from ATP hydrolysis to move molecules against their electrochemical gradients. Uphill work. They fall into 2 main groups. First, the ATP's ion transporters. Within that, you have the P-type KT passes. They get phosphorylated. A phosphate group gets added during their transport cycle. And the absolute prime example, vital for basically all animal life, is the Na plus K plus ATPase. You probably know it as the sodium-potassium pump or just the Na plus AT pump. It's in all our cells.
And its job is fundamental. For every single molecule of ATP it burns, it pumps 3 sodium ions out of the cell and 2 potassium ions in. This isn't just shuffling ions. This constant pumping action is what establishes the crucial ion gradients, especially sodium and potassium gradients across the membrane. These gradients are essential for nerve impulses, muscle contractions, and they also help maintain the cell's volume. It's foundational.
Exactly. And if we want to get into the nitty-gritty like the pros do, the Na plus ACOQ plus ATPase is made of 3 subunits, alpha, beta, and gamma, usually in a 1.1.1 ratio. The alpha subunit is the main workhorse. It has the binding sites for Na plus AK plus A and ATP. It's also where cardiac glycosides like uabane bind, which inhibits the pump that's clinically relevant. Structurally, alpha has a part that crosses the membrane in 3 important domains inside the cell: phosphorylation, nucleotide binding for ATP, and the actuator domain, which helps with the shape changes. The beta subunit helps get the alpha subunit to the membrane correctly and fine-tunes its activity. The gamma subunit also plays a regulatory role, tweaking things like sodium affinity and overall enzyme speed. It's a complex machine. Another type of ATPase ion transporter are the V-type H plus ATPases. You find these mainly in the membranes of intracellular organelles, endosome, lysosomes. That's why they're often called vacuolar H plus ATPases. They pump protons H plus into these compartments, making them acidic. But there's also a plasma membrane version that's important in the kidney for acidifying urine.
Okay, and the second group of ATP-dependent transporters are the ABC transporters, which stands for ATP binding cassette. This is a massive, very diverse family of proteins found everywhere, from bacteria to humans. They all share characteristic ATP binding domains, these cassettes.
And this group brings us straight to a major clinical impact, cystic fibrosis. This is a devastating genetic disease caused by mutations in an ABC transporter gene on chromosome 7. It's the most common lethal genetic disease in white populations, about 1 in 3,000 births. Sadly, death usually comes from respiratory failure. The most common mutation found in maybe 70% of patients is a deletion of just one amino acid, phenylalanine, at position 508. We call it FAVL.
So what's the consequence of losing that one amino acid?
It causes the protein called CFTR, the cystic fibrosis transmembrane conductance regulator, to misfold as it's being made. The cell's quality control machinery in the endoplasmic reticulum recognizes it's faulty and basically targets it for degradation. So very little, if any, functional CFTR protein actually makes it to the plasma membrane where it's supposed to work. Normally, CFTR acts as a channel for chloride and bicarbonate ions. It also helps regulate other transporters like the epithelial sodium channel, ENAC. When CFTR is defective, you get major problems with ion and water transport across epithelial surfaces, especially in the lungs. This leads to abnormally thick, viscous mucus in the airways. This thick mucus gums up the works, impairing the mucociliary clearance system. The little cilia can't effectively sweep out trapped bacteria and debris. This results in chronic lung infections, persistent inflammation, progressive lung damage, and ultimately respiratory failure.
That sounds truly devastating. But you mentioned there's some good news now.
Yes, thankfully, a huge breakthrough. In 2019, the FDA approved Trikafta. It's a combination therapy with 3 drugs: elexacaftor, ivacaftor, and tezacaftor. For patients who have at least one copy of that F50RADL mutation, and that's about 90% of the CF population, Trikafta works remarkably well. It essentially helps the mutated CFTR protein fold better, and increases the amount of functional protein that actually gets to the cell surface. The results have been truly significant, dramatically improving lung function, reducing infections and hospitalizations. It's a prime example of how deep understanding of these transporters leads to life-changing treatments.
That's incredible, a real testament to understanding these molecular machines. And it brings us back to the bigger picture. How are all these transporters regulated? Because membrane proteins aren't permanent fixtures, right? They're constantly being removed and replaced. One key way the cell tags them for removal is by attaching a small protein called ubiquitin to their cytoplasmic part. This tagging is done by enzymes called ubiquitin protein ligases, like the NED4 family.
Exactly. Once it's ubiquitinated, it's like a remove me signal. The protein gets internalized via endocytosis and then broken down by lysosomes or the proteasome. But there's a counterbalance. Deubiquitinating enzymes, or DUBs, which remove ubiquitin. So how long a protein stays active in the membrane depends on this dynamic balance between the ligases adding ubiquitin and the DUBs taking it off.
A great real-world example is how the hormone aldosterone boosts sodium reabsorption in the kidney. One thing aldosterone does is inhibit NED4-2, a ubiquitin ligase. By inhibiting the ligase, it prevents ENA, the sodium channel, from getting ubiquitinated. What happens? More ENA stays in the membrane longer, allowing more sodium to be reabsorbed. Clever mechanism. So while some transporters, like the Na+ pump, are everywhere, many others show really specific expression patterns only found in certain cell types. Think SGLT1 and SGLT2 for glucose transport only in the gut and kidneys, right where you need them. So recapping regulation, you can change the number of transporters, or you can change the activity, rate, or kinetics of the ones already there, like how long an ion channel stays open. Often it's a combination of both.
Okay, so beyond moving individual molecules one by one, cells also need ways to move larger amounts of stuff or even whole particles without compromising that membrane integrity. And that's where vesicular transport comes into play.
Right. Moving bigger cargo. Let's break that down. First, we have endocytosis, which is the general term for bringing substances into the cell by pinching off a piece of the plasma membrane to form a vesicle. There are 3 main types. First, pinocytosis, sometimes called cell drinking. This is a fairly non-specific uptake of small molecules and water dissolved in the extracellular fluid. It's quite prominent in the cells lining capillaries, contributing to fluid exchange there.
Second, Phagocytosis, or cell eating. This is for internalizing large particles like bacteria, cell debris, things like that. It's a really crucial function for immune cells, like neutrophils and macrophages, allowing them to engulf invaders or clean up cellular junk. Phagocytosis is often triggered by receptors. For example, macrophages have receptors that recognize antibodies coating a bacterium. That's called opsonization. Binding triggers the macrophage to wrap around and engulfs the bacterium.
And the third mechanism, receptor-mediated endocytosis, is super specific. It allows the cell to take up large quantities of specific molecules, called ligands, that bind to particular receptors on the cell surface. This involves a whole cast of accessory proteins. Adaptin, clathrin, which forms those characteristic coated pits on the membrane surface, and a protein called dynamin, which helps pinch the vesicle off the membrane. The process usually goes: ligand binds receptor, they fluster in a coated pit. The pit invaginates and pinches off to form a vesicle inside the cell. Then the clathrin coat disassembles and gets recycled, and the vesicle delivers its contents, often by fusing with other organelles like lysosomes. Okay, let's connect this to the clinic. A classic, really important example is cholesterol uptake. Our cells need cholesterol for their membranes, but most get it from the blood. Cholesterol travels in the blood packaged into low-density lipoproteins, or LDLs, often called bad cholesterol. Cells take up LDLs using specific LDL receptors on their surface.
Right. These LDL receptors, once they bind LDL, cluster into those clathrin-coated pits and are brought into the cell via receptor-mediated endocytosis. Inside, the LDL particle is released, and importantly, the LDL receptors are recycled back to the cell surface to grab more LDL. The LDL itself goes to the lysosome, where it's broken down, releasing the cholesterol for the cell to use. Now here's the critical link. Defects in the LDL receptor mean cells can't take up LDL efficiently from the blood. This leads to high levels of LDL cholesterol circulating in the bloodstream. And high LDL is strongly linked to atherosclerosis, the buildup of cholesterol-rich plaques in artery walls, which can block blood flow and lead to heart attacks and strokes. It really highlights how crucial this specific endocytosis pathway is for cardiovascular health.
A very powerful illustration. Okay, going the other way, we have exocytosis, releasing substances from the cell. This happens when intracellular vesicles fuse with the plasma membrane, spilling their contents outside. Exocytosis can be constitutive, meaning it happens continuously, like plasma cells constantly secreting antibodies or fibroblasts secreting collagen components.
Or it can be regulated. This happens in specific cell types. Endocrine cells releasing hormones, neurons releasing neurotransmitters, exocrine glands releasing enzymes like pancreatic cells. In regulated secretion, the vesicles sit ready near the membrane, waiting for a specific signal to trigger fusion. Usually that trigger is an increase in intracellular calcium concentration, K+++D. Although, interestingly, there are a couple of exceptions. Renin secretion from the kidney and parathyroid hormone secretion both seem to be triggered by a decrease in intracellular K++E. Nature always has surprises.
And helping guide these secretory vesicles to the right spot on the plasma membrane are specialized proteins called snare proteins. They ensure the vesicle fuses at the correct location. Finally, there's transcytosis. This is basically a combination of endocytosis on one side of a polarized cell, transport of the vesicle across the cell, and then exocytosis on the other side. It's a way to move substances across an entire cell layer, like transporting antibodies across epithelial barriers in the gut.
Absolutely. This whole system of vesicular transport, it really underscores the cell's incredible logistical capabilities, moving large cargo, keeping things organized, all while maintaining that vital membrane barrier. It's truly cellular engineering at its finest.
Okay, now we're going to dig into the really fundamental driving forces behind all this movement we've been talking about. Because that plasma membrane, with its fatty core, is a big hurdle for many things. Understanding these basic principles is absolutely essential if you want to master this topic like a pro.
Let's start with the simplest, diffusion. At its heart, diffusion is just the spontaneous movement of molecules from where they are more concentrated to where they are less concentrated. It's driven entirely by the random thermal motion of molecules, and the net effect is always to dissipate gradients, to spread things out evenly.
And we can actually quantify this using Fick's first law of diffusion. J equals dA, cycletics. Let's break that down so you really get it. J is the flux, the rate of diffusion, how much stuff moves per unit area per unit time. D is the diffusion coefficient, basically, how easily that specific molecule moves through that specific medium. A is the surface area across which diffusion is happening. AC is the concentration difference driving the movement. And AX is the distance over which that distance exists.
Right. And what affects D, that diffusion coefficient? Well, Smaller molecules diffuse faster. Higher temperatures mean more thermal energy, so faster diffusion. And lower viscosity, a less thick medium, also means faster diffusion. Critically, if everything else stays the same, the diffusion rate, J, is directly proportional to the concentration gradient. Double the gradient, double the rate. Now, applying this to diffusion across a bilayer, that D incorporates how well the molecule passes through the lipid core. We use the partition coefficient to describe how readily a molecule dissolves in the lipid versus the water. If beta is high, it likes the lipid, and diffusion across the bilayer is easier, assuming it can get through the lipid itself.
We can also use a simpler, often more practical equation for diffusion across membranes.
Yeah.
J equals P, Cauchy. Here, J is still flux. P is the permeability coefficient. This is experimentally determined and bundles together all the factors affecting how easily a substance crosses the membrane by a particular pathway, whether that's straight through the lipid or via a channel or carrier. Co and Chi are just the outside and inside concentrations.
And here's a crucial point for true understanding. Simple diffusion directly through the lipid part of the membrane is incredibly inefficient for most biologically important molecules. The source gives some striking numbers. If a cell membrane were just lipids, it would take about 8 minutes for a urea gradient to dissipate. Okay, maybe not too bad. But for glucose or amino acids, 14 hours. And for ions like sodium or potassium, years. This just powerfully hammers home why those specific transport proteins, the channels, carriers, pumps, are so absolutely essential. Life couldn't happen without them speeding things up billions of times. Now, even though it's slow across membranes for many things, simple diffusion is still vital elsewhere. Gas exchange in the lungs, movement of molecules within the cell cytoplasm or between cells in the extracellular fluid.
Yeah, and a great real-world example, exercise and oxygen delivery. At rest, a muscle fiber might be, say, 40 micrometers from the nearest capillary. But during exercise, more capillaries open up, dramatically reducing that diffusion distance to maybe 20 micrometers or less. What's the result? Oxygen and fuels can diffuse to the working muscle much faster, supporting higher metabolic rates. It's physics in action.
Okay, now let's move to a more complex but absolutely fundamental driving force, the electrochemical gradient, or electrochemical potential difference. This quantifies the total force pushing a molecule across a membrane. And it's key because it has two components. First, the chemical potential difference driven by the concentration gradient we just talked about, pure diffusion. Second, the electrical potential difference. This only matters for charged molecules, ions, and only when there's a voltage difference across the membrane, which there almost always is in cells, the membrane potential, VM.
So the overall equation looks like this. AXRT-NAMXI plus VXSVM. Let's unpack that quickly. R is the gas constant, T is absolute temperature, the NXSI part is the chemical gradient term, ZX is the charge of the ion, F is the Faraday constant, and V is the membrane potential. So for something uncharged like glucose, that second electrical term is zero, Z0, only the concentration gradient matters. But for an ion like potassium, K plus C, Z plus 1, both the concentration difference and the membrane voltage push or pull on it. And this leads us straight to the incredibly important Nernst equation. This is derived from the electrochemical gradient. It calculates the Nernst equilibrium potential, XX, for a specific ion. X is the membrane voltage at which the electrical force exactly balances the chemical force for that ion, resulting in no net movement. It essentially tells you how much electrical energy is stored in that ion's concentration gradient, expressed in millivolts.
Exactly. And for practical purposes, working at body temperature, 37 degrees C, we often use simplified forms. X equals 61.56 log 10, that's aside, or sometimes X equals 61.5 log 10 celsius. The key insight here is the magnitude. For a univalent ion, charge plus 1 or METF 1, a 10-fold concentration difference across the membrane is equivalent to 61.5 millivolts of electrical potential energy.
Yeah.
A 100-fold difference, that's 123 millivolts, huge potential. For a divalent ion like Kl++ Iz plus 2, a 10-fold gradient is worth about 30.7 millivolts. So going back to potassium, its concentration gradient typically pushes it out of the cell. The membrane potential, usually negative inside, pulls it in. The Nernst potential tells you the voltage where those two forces balance. The actual membrane potential determines the net driving force and direction of movement.
Okay, so putting the gradients and energy together, what does this mean for how things actually move? Is the cell spending energy or not?
That perfectly sets up the distinction between active versus passive transport. These are the cell's two fundamental energy strategies for transport. Passive transport is when the net movement of a molecule across the membrane happens in the direction predicted by its electrochemical gradient. It's downhill movement going with the combined chemical and electrical forces. This does not require the cell to directly expend metabolic energy at that moment. Examples are water moving through aquaporins, ions flowing through open channels, or glucose moving via a GLUT uniporter if glucose is higher outside.
And the opposite is active transport. This is when the net movement occurs opposite to the direction predicted by the electrochemical gradient. It's uphill movement going against the gradient. And this always requires the cell to provide energy, usually from ATP.
Right. And active transport itself is further divided into two critical types. First, primary active transport. Here, the transport process is directly coupled to the hydrolysis of ATP. The energy released from breaking that ATP bond is used right there by the transporter to pump the molecule uphill. Our main examples are those ATP-dependent transporters, the Na plus K plus ATPase, the various H plus ATPases, proton pumps, and the ABC transporters we discussed. They are the direct energy consumers. Second, we have secondary active transport. This is a bit more indirect, but incredibly important. It occurs via those coupled solute carriers, symporters and antiporters. In this clever system, the downhill movement of one molecule, following its own favorable electrochemical gradient, provides the energy to drive the uphill movement of another molecule against its gradient. Think of the Na plus Na glucose symporters or the Na plus NHH plus antiquarter. The strong inward electrochemical gradient for Na plus, maintained by the primary active Na plus Na pump, is harnessed to pull glucose in against its gradient, or push H plus out against its gradient. It's using the energy stored in one gradient to build another.
Let's look at that clinical example again. Glucose transport in the gut and kidney tubules. This is classic secondary active transport. Glucose is absorbed into the epithelial cells from the lumen using sodium-glucose-linked transporters, SGLT1 and SGLT2. They use the downhill movement of Na plus into the cell to pull glucose in with it, even if glucose concentration inside the cell is already high. Now there's a difference. SGLT2 transports one glucose with one Na plus NAP. Using the typical Na plus gradient and membrane potential, it can concentrate glucose inside the cell about 100-fold compared to the outside. So if internal glucose is 2 millimem, it can pull glucose in until the external concentration drops to about 0.02 millimeter. Pretty good.
But SGLT1 is even more powerful. It transports one glucose molecule coupled to two NASPL plus ions. By harnessing the energy from two sodium ions moving downhill, SGLT1 can generate a much steeper glucose gradient, nearly 10,000-fold. Same scenario, if intracellular glucose is 2 millilem, SGLT1 can continue pulling glucose in until the lumen concentration is down to an incredibly low 0.0002 millilem. It can virtually scavenge every last bit of glucose. This really demonstrates the power of coupling stoichiometry and secondary active transport. It's how our bodies ensure we don't waste valuable nutrients.
That's fantastic. It shows how this intricate understanding of energy use is just fundamental to how our bodies maintain these critical balances, absorbing what we need, getting rid of waste, all against steep odds sometimes. Real evolutionary efficiency. Okay, let's broaden our view now to the solutions themselves, the physiological fluids that bathe our cells. Understanding their properties is just as crucial. Our sources highlight several key concepts. Foundational ideas like molarity and equivalence are obviously essential for working with physiological solutions, but they aren't detailed in this specific material, so we'll focus on what is covered here for our deep dive.
Right, so let's start with osmosis and osmotic pressure. We know water movement across cell membranes is passive. What drives it? The difference in osmotic pressure. Think of a semi-permeable membrane lets water pass, but not solutes. Water moves from the side with higher water concentration, lower solute, to the side with lower water concentration, higher solute. We quantify this driving force, the osmotic pressure, using Van Hoff's law, Ohm's NCRT pi, is osmotic pressure, N is the number of particles the solute associates into, e.g. L is M1 for glucose, N approx 2 for NaCl. C is the molar concentration. R is the gas constant, T is absolute temperature. The key takeaway, osmotic pressure depends on the number of solute particles, not their size or charge. More particles, higher osmotic pressure. Now, a really critical distinction, often confused, is osmolarity versus osmolality. Osmolarity is the number of osmoles of solute per liter of solution. Because volume changes with temperature, osmolarity is temperature dependent. Osmolality is the number of osmoles of solute per kilogram of solvent, usually water. Because mass doesn't change with temperature, osmolality is temperature independent. For biological systems, osmolality, measured in osm kg H2O, is strongly preferred because it gives a stable measure regardless of temperature fluctuations.
Okay, then there's tonicity. This term is defined purely by its effect on cell volume when a cell is placed in that solution. An isotonic solution, no change in cell volume. A hypotonic solution, cell swells. A hypertonic solution, cell shrinks. And here's the absolute key difference from osmolality. Tonicity takes into account whether the salt particles can actually cross the cell membrane. Osmolality just counts particles.
Exactly. The classic example makes this crystal clear. Sucrose versus urea in red blood cells. You can prepare solutions of sucrose and urea that have the exact same osmolality, they're isosmotic, but put red blood cells in the isosmotic urea solution. They swell up and burst. Why? Because urea is an ineffective osmole. It crosses the cell membrane fairly easily, so enters the cell, increases the intracellular solute concentration, water follows, and boom. Put the cells in the isosmotic sucrose solution. They maintain their volume. Sucrose is an effective osmole. It cannot cross the membrane. So the external sucrose balances the internal solutes, preventing net water movement. To quantify this effectiveness, we use the reflection coefficient. Sigma ranges from 0 for freely permeable solutes like urea to 1 for completely impermeable solutes like sucrose. The effective osmotic pressure is actually Bureau's CRT. This sigma factor is crucial for predicting tonicity.
Good distinction. Next, oncotic pressure, sometimes called colloid osmotic pressure. This is the osmotic pressure generated specifically by large molecules, primarily proteins like albumin in places like blood plasma. Our source mentions it doesn't always follow Van Hoff's law perfectly, often being higher than predicted, possibly due to protein interactions or shape. But even though the absolute value, around 26-28 millimeter of Hg in plasma, seems small compared to total osmotic pressure, it's hugely important for regulating fluid movement between capillaries and the surrounding tissues.
And finally, specific gravity. This is a simpler concept. The weight of a volume of solution compared to the weight of an equal volume of pure water. Distilled water has a specific gravity of 1.000. Biological fluids containing solutes are denser, so their specific gravity is greater than 1. Human plasma is typically around 1.008 to 1.010.
Clinically, urine-specific gravity is often measured as a quick check of the kidney's concentrating ability. Higher specific gravity generally means more concentrated urine, correlating roughly with osmolality. But, and this is key for that elite professional understanding, the relationship isn't perfect. Specific gravity depends on both the number and the weight of solute particles. So if a patient has received intravenous contrast dye for an X-ray, which has very large, heavy molecules, their urine-specific gravity might shoot up to 1.040 or 1.050, even if the actual osmolality hasn't changed much. You have to consider what is contributing to the density. It's a critical detail. Wow. We have covered a ton of ground today. We started deep inside the eukaryotic cell, explored its intricate machinery, then moved to that dynamic plasma membrane. We unpacked the whole network of transport proteins, delved into the driving forces like diffusion and electrochemical gradients, clearly distinguished active versus passive transport, including those clinical links like CF. And we finished by clarifying those crucial properties of physiological solutions: osmosis, osmolality, tonicity, oncotic pressure. You really should have a much deeper, interconnected view now.
Absolutely. To really nail down the core takeaways for mastery, remember, the plasma membrane is that dynamic lipid bilayer with embedded proteins, orchestrating transport via aquaporins, ion channels, solute carriers, and ATP-dependent transporters. Movement across it is driven by chemical gradients and electrical potentials. The electrochemical gradient quantifies this. ATP-dependent transporters are key for establishing these gradients. Water follows osmotic gradients. Transport is either passive, downhill with the gradient, or active, uphill against it, requiring energy. Active is split into primary, direct ATP use, and secondary, using another gradient's energy. That framework is essential.
You definitely now have a deep understanding of these fundamental principles, the kind that elite professionals in physiology and cell biology rely on every day. It's just fascinating how these microscopic events dictate so much of our overall health.
And as we think about how incredibly precise and regulated all this cellular transport is, it does make you wonder, doesn't it? Beyond the major diseases like cystic fibrosis, what about more subtle variations or dysfunctions in these transport mechanisms? How might common health challenges maybe related to hydration, nutrient absorption, electrolyte balance be influenced, perhaps quite significantly, by the fine-tuning and efficiency of these very processes we've discussed today in ways we're maybe just starting to uncover?
A great question to ponder. Keep digging deeper, stay curious, and we'll see you on the next deep dive.